The current widely used photosynthesis model was developed 40 years ago.
Great.
But science has been advancing, and the requirements for photosynthesis modeling have broadened tremendously. Here, I will give a brief summary on our effort to modernize photosynthesis modeling. We are doing this by extending from a traditional focus on biochemistry of photosynthesis to a coupled modeling of photophysics, photochemistry, and biochemistry. Along the way, we not only have a more complete photosynthesis model, but also are able to explain some enduring mysteries in nanoplants. Photosynthesis consists of a sequence of events that occur at drastically different timescales, covering at least 19 orders of magnitude in time. Typically, photosynthesis is divided into two stages of broad reactions, the light and carbon reactions. This two-stage separation, however, groups the light reactions too broadly. From a system modeling point of view, it is much more insightful to divide photosynthesis into three stages, or three eras of reactions, according to Martin Kamen, based on the characteristic timescales of consecutive photosynthetic events, photophysics, photochemistry, and biochemistry. In the Kamen division of photosynthesis, the light reactions are further partitioned into the photophysical reactions and photochemical reactions because these two groups of reactions are spatially separated, follow different laws, and operate at vastly contrasting timescales. The photophysical reactions occur at timescales of pico to nanoseconds. The photochemical reactions occur at timescales of nano to milliseconds. The biochemical reactions occur at timescales of milliseconds to minutes. These three eras are then followed by the eras of physiology and ecology, which occur at longer timescales. The photophysical reactions cover the stages of light harvesting in the antenna complexes and the transfer of excitation energy to the reaction centers of photosystem 2 and photosystem 1. These reactions are governed by physical principles such as quantum mechanics, energy conservation, and Foster resonance excitation energy transfer. The photochemical reactions cover the subsequent electron transport, including water splitting in the oxygen evolving complex, charge separation, acquiring of electrons by acceptors in the reaction centers, transfer of electrons to mobile carriers either within the lipid bilayer core of the liquid membrane, lumen, or stroma, and to the eventual acceptor NADP+ to produce NADPH in the stroma to be used in the biochemical reactions. This stage of reactions also includes electron exchanges within the cytochrome B6F complex, proton pumping across the liquid membrane, and ATP synthesis. The photochemical reactions are of the reduction oxidation nature and governed by thermodynamics, electrochemistry, and chemiosmotic theory. The ATP and NADPH produced at the end of photochemical reactions are used in the biochemical reactions to reduce CO2 to form sugar. Current photosynthesis models have not represented the three stages of photosynthesis equally well and have not been parameterized statistically adequately, in my opinion. The FVCB model dominates photosynthesis modeling. It is a biochemical model. It has no explicit representations of photophysics or photochemistry. It is typically parameterized with ACI curve measurements made at a constant saturating light. A model parameterized in this limited way is then used to make predictions for conditions of variable light, which violates a basic principle of model application. We need a more complete model of photosynthesis for a lot of applications, for example, to improve carbon cycle and land surface modeling, and to provide guidance for genetic improvement of photosynthetic efficiency. Photosynthesis is extremely complex. If you are ambitious, you can easily end up with a model that has hundreds or even thousands of equations with lots of unknown parameters. Our goal is more modest. We want to have a model that is capable of broad applications, by which we mean that the model should have similarly rigorous representations of photophysics and photochemistry as the biochemistry in the FVCB model, and all model parameters can be obtained from common leaf-level measurements such as PEM fluorimetry and gas exchange. This goal led us to settle on this structure of the model. The photophysical model predicts energy dissipation pathways from solar energy input and regulatory mechanisms. The photochemical model describes how the structural properties of electron transport chain constrain the relationship between electron transport rate and the redox state of protein complex and mobile electron carriers. The biochemistry remains the same as in the FVCB model, except that we now attempt to account for ATP and NADPH consumptions in synthesis, in addition to CO2 assimilation and photorespiration. Key feedback controls from biochemistry to photochemistry and from photochemistry to photophysics are also represented. The photophysical model is derived from the principle of energy conservation. It expresses the linear electron transport rate from photosystem 2 to photosystem 1 as a function of absorbed light partitioned to photosystem 2, non-photochemical quenching (NPQ), and the fraction of open photosystem 2 reaction centers denoted by QL. Phi PS2 max is the maximum photochemical quantum yield of PS2. Energy dissipation rates in fluorescence, regulated and unregulated heat can be similarly formulated as a function of NPQ and QL. Here we assume a lake model connectivity of photosynthetic units. A Potter model version can also be derived. The complexity of the photochemical model depends on how the redox states of the reaction centers are classified and how many events along the electron transport chain are represented. Here, this equation is derived by separating the photosystem 2 reaction centers into an open or closed state, which is consistent with the typical PEM fluorimetry applications. Key processes represented in the model include redox reactions between the acceptors of photosystem 2 and the mobile electron carrier plastoquinone, and between plastoquinone and the cytochrome B6F complex. The light-induced thylakoid swelling and shrinking, the dependence of redox reactions on temperature according to the Markos theory of electron transfer in proteins, and the redox poise balance between photosystem 2 and the cytochrome B6F complex, which is under the photosynthetic control. It can be confusing as to why we have two models that relate the fraction of open PS2 reaction centers to linear electron transport rate. In fact, this was one of the reasons why our manuscript on photochemical modeling was reviewed by at least 8 referees and rejected by 2 plant science journals, each time due to a stubborn referee. It took us tremendous efforts to explain to the referees what we are trying to model and why we are doing this modeling. It is important to understand the differences between the photochemical model and the photophysical model. The photophysical model predicts competition for excitation energy within the light harvesting antenna complexes. It governs processes that occur before the excitation energy has been delivered to the reaction centers. It requires an assumption about the photosynthetic unit connectivity, either lake model or Potter model. It reflects controls of regulatory mechanisms on energy dissipation pathways. In contrast, the photochemical model predicts the relationship between linear electron transport rate and redox states of protein complexes. and electron carriers along the electron transport chain. It governs the processes that occur after the excitation energy has been delivered to reaction centers and charge separation has been initiated. It is independent of photosynthetic unit connectivity, and it reflects the structural properties of the electron transport chain. Note that the photophysical and photochemical models together do not form a closed system because we have two equations, but with three unknowns, JPS2, NPQ, and Q. There are three potential solutions to close the system. The first is to develop an independent model to predict NPQ from environmental conditions. The second is to develop an independent model to predict Q from environmental conditions. The third solution is to use a model of biochemical reactions of photosynthesis to independently predict JPS2. All three solutions could work, but here we focus on the use of biochemical model of photosynthesis because this solution allows us to predict variables of both light and carbon reactions. But before we talk about how to couple the photophysical and photochemical models with the biochemical model, we need to realize that JPS2 is the whole chain electron transport rate, and some of these electrons are used in processes other than CO2 reduction, such as nitrate reduction, Mehler reactions, and VOC synthesis. If we compare the electron transport rate calculated from PEM fluorimetry, which is the y-axis and treated as JPS2, and the electron transport rate calculated from CO2 assimilation denoted as JA here, with the FVCB model, which is the x-axis, and treated as the rate that actually supports photosynthesis, we see that JPS2 tends to be higher than JA. We therefore have to expand the traditional FVCB model to account for alternative electron sinks for the coupling of the three stages of photosynthesis. On this slide, the left side is the traditional FVCB model. The right side are the new equations we derived for modeling alternative electron sinks. Key assumptions are the following. The ratio of ATP to NADPH is balanced for photosynthesis. A fixed fraction of ATP G other is used to support alternative stromal reactions other than photosynthesis. A dynamic fraction of NADPH G total is used to support pseudocyclic electron transport, such as Mehler reactions and synthesis of amino acids, lipids, and VOC. State transition is linearly related to the reduction level of the mobile PQ pool. The PS2 to PS1 ratio of photochemical quantum yield Kc is constant, allowing the fraction of cyclic electron transport around the PS1 to be determined with the state transition. LiveWeb has collected a large dataset of joint PEM fluorimetry and gas exchange, which was used in testing our models. The dataset includes species from the tropics to boreal, and from lianas to trees to grasses to crops, covering both C3 and C4 species. The photochemical model fits the observed JPS2Q relationship well. Conventional thinking is that state transition is not significant for higher planes, but that thinking is not consistent with what we have found. Without explicit representation of state transition, the model cannot fit light and CO2 response curves simultaneously, as demonstrated by the difference between plot A and B. Also, the calculated JPS2 differs between with and without state transition. One of the exciting aspects of scientific exploration is that you never know for sure what your research will lead you to. During our effort to develop a complete model of photosynthesis, we found explanations to some of the deepest mysteries in higher planes. Granite stacks are universally present in the lilocoids of higher planes, but are almost completely absent in other oxygenic photosynthetic organisms. Protein complexes required for coordinated photon harvesting and photosynthetic electron transport are separated distantly in lilocoids by a path subject to macromolecular blocking for diffusion of electron carriers. Photosystem 2 is more frequently photodamaged than photosystem 1. Shade leaves in lower canopy have taller granite stacks than sun leaves in the upper canopy. The capacity for diverting electron flow to oxygen is considerably smaller in higher planes than in cyanobacteria and algae. Nanoplanes emerged from Charophyta, not from other algae groups. These phenomena are so difficult to explain that some people think we should just take them as granted. They just happen that way. But is this true? Here is a 3D model of thylakoid membrane of higher planes, reconstructed from electron tomography. Granite stack looks like a column of coins and is wrapped around by stroma lamina. The diameter of a granite stack is about 400 nanometer. The width of stroma lamina is about 200 nanometer. The internal space of each granum is called a lumen, with a thickness of 4.7 nanometer, and the membrane has a thickness of about 4 nanometer. The gap between two neighboring grana is called a partition. It has a thickness of about 3.6 nanometer. This ball is called plastoglobulus, which acts as a lipid reservoir. Here is another 3D model of thylakoid of higher planes. PS2 is mostly located in granite stacks. PS1 and ATP synthase are mostly found in stroma lamina. Cytochrome B6F complex is evenly distributed between granite stacks and stroma lamina. There are two types of mobile electron carriers that work inside the thylakoid. Plastoquinone PQ diffuses in the lipid bilayer core to carry electrons from photosystem 2 to cytochrome B6F complex. Plastocyanin PC diffuses in the lumen to carry electrons from cytochrome B6F complex to PS1. Both PQ and PC must negotiate tortuous multimedia paths to deliver electrons. Granite stack membranes are heavily packed with PS2 and light harvesting complexes, blocking diffusion of PQ. Lumen is narrow relative to the size of PC and subject to blockage by the OEC of PS2. Neither PQ nor PC could diffuse freely in their respective domains. The bipartite architecture of thylakoid of higher planes is apparently at the expense of electron transport efficiency. If that's the case, then one might ask why evolution selects an apparently unnecessary, inefficient bipartition. Granite stacks are not necessary for oxygenic photosynthesis to occur. Cyanobacteria and most algae do not have such a system. Charophyta is an exception. Nanoplanes embryophyta emerged from within Charophyta, perhaps from terrestrial unicellular charophytes. Although the bipartite system is not essential for oxygenic photosynthesis to occur, it may be essential for embryophyta to survive on land. It turns out thylakoid swells in light and shrinks in dark. As the light shines on chloroplast, thylakoid swells. The swelling is due to the expansion of volume of lumen, both in the granite stacks and in the stroma lamina. The narrow space between two neighboring granite disks, known as partition gap or stromal gap, is squeezed as the granule lumen expands. In the darkness, thylakoid shrinks. Here are some more detailed light-dark comparisons in the thylakoid dimensions. The repeat distance is the distance between the centers of two neighboring stromal gaps. This distance increases as light is turned on. Meanwhile, the stromal gap gets narrower. The Newman gap gets wider. Plot D shows a schematic contrast between dark and light adapted granite. You might ask, can thylakoid burst if it swells in the light? Guard cells swell, but don't burst because plant cells have rigid walls. Chloroplasts have aminos, but don't have rigid walls. Thylakoid does not burst because of an organelle called plastoglobuli. Plastoglobuli are often found to be attached to the granule margins. They are formed by a membrane lipid. The role of lipid reservoir to support the thylakoid swelling and shrinking by feeding or taking up lipids as necessary in a carousel fashion, like kids playing merry-go-round. So what functions do thylakoid swelling and shrinking have on photosynthesis? It turns out thylakoid swells to allow electron carriers to move more freely. In the darkness, thylakoid shrinks to confine electron carriers tightly in place. Proper representation of thylakoid swelling shrinking is important for modeling photosynthetic electron transport. This counter figure compares the measured and fitted relationships between the linear electron transport rate and the fraction of open PS2 reaction centers for different species. The left column is the model performance when the thylakoid swelling shrinking is represented. The right column is the model performance when the thylakoid swelling shrinking is pretended not to happen. This black-white figure compares the modeled and fitted linear electron transport rates for different species. Again, the left column shows the result for the model with thylakoid swelling enabled, while the right column shows the result with the thylakoid swelling shrinking disabled. The model performance deteriorates when the thylakoid ultrastructural dynamics are not explicitly represented. These results beg the question, what evolutionary benefits does thylakoid swelling and shrinking have for higher plants? We proposed the Bellows theory to explain this mystery. The Bellows theory suggests that granite stacks and guard cells to a leaf are like bellows and air button to an accordion. Granite stacks exert ultrastructural control on photosynthesis through thylakoid swelling and shrinking to ensure electron transport through the electron transport chain is in balance with CO2 supply through stomata. The thylakoid swelling and shrinking is induced by osmotic water fluxes in coordination with varying stomatoconductance and a turgor of guard cells. Granite stacks protect photosystems from photooxidative damage and represent a dry, high irradiance adaptation of photosynthetic machinery to improve fitness in challenging land environments. Having granite stacks, Charophyta may have paved the way for higher plants to successfully conquer lands. The Bellows theory unifies many well-known phenomena of thylakoid structure and function of higher plants. For more information on Bellows theory, please read this paper. To conclude, we have developed a coupled model of photophysics, photochemistry, and biochemistry of photosynthesis to predict a wide range of variables of interest to plant scientists and ecologists. Energy dissipation pathways, including redox states of protein complexes and electron carriers along the electron transport chain, state transition, linear, cyclic, and pseudo-cyclic electron transport, CO2 assimilation. The complete model enables the use of both PEM fluorimetry and gas exchange measurements to improve photosynthesis and carbon cycle modeling. And this model can be used to determine how components of the photophysical, photochemical, and biochemical reactions can be bioengineered to improve the overall efficiency of the photosynthetic machinery. Here is a little animation and some accordion music for you to enjoy.